So genome reports for the ERGA community. First of all, just as a kind of brief reminder of what we mean when we're talking about a genome report. And this is very much a technical publication that describes, all the steps that were taken to produce the reference genome, from sampling through sequencing, assembling, and in many cases, annotating as well. And it's following a very standardized format and structure so that readers can very quickly and easily understand the quality of the reference resource and how it was generated. So basically, just to distinguish it from, let's say, a paper where you do downstream analysis and you have some evolutionary results, this is not an analysis and results type paper. This is a data type paper. And why are we encouraging the publications of these genome reports? On the one hand, it's to really allow and encourage the proper recognition of everyone who contributed to the production of the reference resource in a citable manner. This is because if the genome itself is only kind of published as part of a bigger study, A, the methods are usually very short and so not reproducible, and B, some of the people involved in the early steps of actually sampling and doing the extractions, etc., don't necessarily get the recognition that they deserve in those kind of more science and results facing papers compared to these data papers. They are also, of course, with a structure and the details supporting open and reproducible science. That, as I said, can sometimes be overlooked in your classical research manuscripts where perhaps the journal says, your method section is too long, please delete half of it. And then all of the methods that you've collected about how you produced the reference resource disappear and no one can ever reproduce it. It also helps to speed things up because as soon as the data are ready, as soon as the assembly is ready, we can write the genome report and publish it. You don't have to wait for months and sometimes years for all of the downstream analyses to happen. So this is why we encourage the publication of genome reports. Probably most of you will be familiar with the collection of Wellcome genome notes published at the Wellcome Open Research Gateway, which behind it is the F1000 publishing office. And these are all species that are sequenced by the Darwin Tree of Life project, which is great. And there's a fantastic collection there, and you can kind of get a sense of how useful these are in the standardized format and the citability. They also go straight out as a preprint, so you don't have to wait for the full peer review. That can happen later. However, of course, these are limited to researchers who are funded by the Wellcome Trust. It's natural the Wellcome Trust is funding this thing, so it's for Wellcome Trust researchers. So basically, we wanted to create a similar kind of collection that is open for all the ERGA community, not just those funded by the Wellcome Trust. And so we wanted it to be accessible to all. And at the same time, we didn't want it to be too restrictive in terms of being the only place where our community is able to publish these genome reports. So we know that the community is publishing genome reports in a variety of different places. There's some examples, some journals there. And so we wanted to find a solution that could bring them all together. We also realized that if we can put together some guidelines and a platform to make it easier to actually create one of these standardized genome reports, then we can minimize the effort of the community required to produce one, which would then increase the number of these that we can actually produce and publish. And so after some searching high and low, we identified the Pensoft Research Ideas and Outcome, so RIO journal, as a solution. Because on that platform, they allow both preprinting and publishing of publications in a standardized format, which we have been working with them to create to meet the specifications of an ERGA genome report. Importantly, it also allows adding to this collection genome reports that are published elsewhere. So that means there's no restriction on the ERGA community. If you want to publish in GigaScience, you can publish in GigaScience, but we can still include that publication in the collection in a relatively easy step that I'll explain in a couple of minutes. So this is what the website looked like for the RIO journal. And to find the ERGA genome reports, you just click on the topical collections and scroll down a bit. and then you will find this permanent collection, the European Reference Genome Atlas ERGA Community Genome Reports. And it's currently edited by myself, Camilla, and Claudio, because to set this up was funded by the Biodiversity Genomics Europe project. But obviously in the future, we could think about expanding this list of editors and updating it. So there's a description of kind of what the collection is all about, who funds the collection startup, and what kinds of publications we're looking for in terms of these structured genome reports. So the beauty here is that we're looking at 3 different ways to accommodate the variety of possible sources of genome reports from the ERGA community. So the first one is simply add your genome report that has been published elsewhere to the collection. And here we're working on the guidelines to help the community do this as we're testing the first ones coming through. But it's a fairly simple process. You add document to collection, you confirm that you have the consent of all of the authors, you import or add the metadata that describes your paper, so the names and emails of your authors. You hit the submit request, the editors check that it meets the requirements, and if it's accepted, then it'll appear in the collection linked to the source publication. Alternatively, you can publish an author formatted PDF as a preprint on the collection, and importantly, this is totally for free. So, you know, ERGA communities wouldn't incur any cost to publish a preprint in this same collection. And there again, you start the manuscripts, again confirming that you have consentable authors. There you have to add the metadata yourself, because of course it's not yet published somewhere else. Submit the request, we check that it meets the requirements, and then it can appear in the collection as a preprint. And you'll see there are already quite a few there in the collection as preprints. Then finally, the last option, you can actually use their publication creation or their document writing platform called the ARFA platform to create your genome report and submit it for peer review. So you start the manuscript, you write and submit it using the ARFA tool. This goes then through to the editors, who then send it out for peer review. With 2 at least positive reviews, we can then accept it, and then it'll appear in the collection as a peer-reviewed publication. Now, this version of all of the options that we have available does incur a small article processing charge, but it's really small compared to most other journals. So we're talking in the region of about 300 euros. So, you know, if you want to go all the way through to publication rather than just stopping after pre-printing, at least it's a fairly affordable option. And I guess as things kind of progress and we see how the community is using this resource, if there are many of us, we might be able to go back to the journal and kind of try to negotiate to even decrease that a little bit further. So those are the 3 avenues, if you like. And this is kind of what it looks like on the website. As I said, there's the option to start a manuscript, or there's the option to add a document to the collection. And note here that adding a document to the collection is completely free to the ERGA community. So the description obviously is kind of short on the website. So what we've been working on recently is a more detailed list of requirements and recommendations. So this is so that everyone in the ERGA community is aware of what is expected of a genome report to be acceptable for inclusion in this collection. And so there's a link on the page, and here I'm just showing a couple of screenshots. Developed by Tom Brown with input from the executive board and the committees. And here, I'm not going to go through all the details now, but this is really just to point you in the right direction so that if you're thinking of writing a genome report for your reference assembly, you know where to look for the recommendations and the requirements. And essentially, on the requirement side of things, we're looking for to add a manuscript published elsewhere to the collection. We need a DOI, digital object identifier, to the genome report. The data paper should include detailed methods descriptions, detailed sample metadata. It should have the accessions to the data sets that are sitting at an INSDC or the European Nucleotide Archive. Importantly, the bioproject needs to be linked to one of the ERGA umbrella bioprojects, and there's instructions in the guidelines on who to contact to get that to happen. Importantly, because we are ERGA, European Reference Genome Atlas, the species should be European species. So we know that lots of ERGA members are also sometimes working on exotic species, but here we really wanted to keep it a European Reference Genome Atlas. Then a brief biological description of the species, plus, of course, the detailed genome quality metrics, and finally, the full acknowledgement of all the contributors. Now, this is a little bit kind of difficult to check each and every single one of these things in detail. So it's going to be a cursory review to make sure that what is being submitted, at least on the face of it, looks like it is ticking all of these boxes. Then apart from the the requirements, we also have a couple of recommendations. We would recommend that it's published open access somewhere, but we recognize that that's not always possible. We also recommend that it's meeting the EBP standards, noting that there are some exceptions where taxa or genomes are not quite meeting the quality metrics just because of the biology of the system in which we're working. We also, really strongly recommend that the genome report and all the metadata and data workflows, etc., are adhering to FAIR principles in as much as possible. And finally, we recommend that the genome report should, wherever relevant, mention, for example, in the acknowledgement section, any support that has been received from ERGA. So whether that's the use of SOPs or guidelines or even interactions with the committee, that somehow appears, for example, in the acknowledgement section. So this is what the preprints look like on the collection. These are two examples coming from the Biodiversity Genomics Europe project. So here you can see they are labeled as published in ARFA preprints. So these ones we have formatted, uploaded, added the metadata, and then it's been accepted into the collection as a preprint. And you can, when you click on one of them, it's clearly marked as a preprint posted on the ARFA preprint server with all the typical information you would expect of a typical publication. So the PDF that was submitted and then all the metadata of the authors involved. I'm not going to go into a huge amount of detail today about why this is so important, but I will maybe try to put this link in the chat afterwards, which is a paper published on RIO explaining the importance of improving the way we publish to be much more machine readable, especially when it comes to methods, accession numbers, etc. So we can make our data descriptions much more machine readable, which when you think about the numbers of genomes that we're producing is really important for the future. So I will not go into too much detail there, but I'll drop the link in the chat afterwards. So in terms of identifying examples from the community already, we've had a little bit of a look around and we've identified quite a few. So these are some of them coming from the ERGA pilot, some of them just coming from other ERGA members, again, where they've mentioned ERGA in the acknowledgements or the ERGA pilot in the acknowledgements, and they're published in various different resources. So this is how we started to get going with adding genome reports that are published somewhere else to the collection. And so finally here, we just have an example of two hair genomes and the violet carpenter bee. So these are both coming from the ERGA pilot. And you can see the top one here published in Peer Community Journal, as well as the bottom one here, Peer Community Journal. And this one was the violet carpenter bee published in Heredity. So we are very easily able to quickly bring together all of the outputs of the ERGA community, no matter where they're sitting, so without any restrictions on where you wish to publish. So this is where we are today. And I guess now the invitation is there to begin to contribute to the collection, either by submitting genome reports that have been published elsewhere to be linked in this collection, or working with the IT and Infrastructure Committee to kind of format your PDF for posting as a preprint in this collection, which would be at zero cost to the community. Also beginning to explore how to create and then submit for peer review publication also within this collection for obviously that small article processing fee. So I think we've made a lot of progress, and now let's see, as the community starts to use this, we will continue to develop the guidelines to make it easier to follow, because obviously there's 3 different routes here. Which is the best one for you may not be so obvious at the beginning, but we hope that as people start using it, it'll become more and more obvious. So with that, I will stop sharing and open the floor to any questions about the genome reports and this platform.